Hello and welcome to another video here for our genetics unit. Today, what we're going to be discussing is the concept of blood typing. Now, blood typing is actually a co-dominant trait. So what we're going to be looking at here is what blood typing is, what the different blood types are, and how to do a couple of problems relating to blood type itself. So without further ado, let's get started. Now, the classic example of co-dominance in humans is our blood type. For example, the gene that controls the A, B, or O blood type codes for an enzyme that makes glycolipids on the blood cell. So as you can see here, we have different lipids or proteins found on the surface of the blood. The two alleles A and B are codominant, whereas the third allele, O, is recessive to both A and B. Now this makes sense because as you can see here, if you are blood type A, you only have the A antigen. If you're blood type B, you only have the B antigen. If you are A and B, so if you have the AB blood type, you have both the A and B antigen found on the surface of your blood, which makes sense because these two are co-dominant of each other. Both are dominant. Meanwhile, if you have O type, the only pure recessive type, you don't have any antigens on your blood at all. Now, this is going to come in handy later when we discuss how to go ahead and the process of getting blood donated, transfusions, things of that nature. So let's get to that. So the possible genotypes for blood. If you have the phenotype A, now remember we have three possible alleles. We have A, B, and O. So the combination for the A blood type is AA or AO, because remember, A is dominant to O. If you have the B blood type, your combination or your genotype can be BB or BO, because as we said before, A and B are both dominant to the O-type blood. If you have the phenotype AB, there is only one combination you can have, which is 1A and 1B. And finally, if you have the phenotype O, the only combination you can have is OO, because this is the recessive genotype as well as the recessive phenotype. So the only combination for O-type blood is OO. So the ABO blood system, here's what this all kind of means. Your blood type is determined by what A and or B proteins are on the surface of your blood. So as you can see here, let's go ahead and use red, seems appropriate. So if you are A-type, that means you have the A proteins on the surface of your blood cells. Now, what you also have are the B antibodies, which means that if anything with the B protein is found inside your blood, these antibodies are going to attack it and treat it as a threat. So, in other words, if you're A-type and you got a donation from anybody that had a B-type protein on their blood, your body wouldn't take it. Meanwhile, B-type blood has the B proteins on its surface, and inside the blood it has the A antibodies. So if it found anything with the letter A on it, on the surface of the blood, it would attack it. Continuing with that, AB blood type has both the A and B antibodies on it. Meanwhile, the A and B proteins. As you can see here, it has no antibodies in it, meaning that AB is known as the universal acceptor. So the universal acceptor means that it can take any type of blood. If you're type AB, you can accept donations from A-type, B-type, AB-type, or O-type. And finally, if you're O-type, that means you have no proteins on the surface of your blood cell. However, the trade-off for this is that you have both the A and B antibodies in your bloodstream. Now, if you are the O-type blood, you're the universal donor because you don't have any proteins on the surface of your blood, meaning you can donate to anybody. However, there's kind of a backside to this, which means that you have both the A and B antibodies in your bloodstream, meaning that if you go ahead and decide to get a donation from A-type, B-type, or AB-type, your body will reject it. So if you are O-type, the only type of blood you can receive is O. Now, blood tests, ways to figure out what type of antibodies or what blood type you have. The way this is carried out is we take 2 samples of the blood and then add the anti-A antibodies as well as the anti-B antibodies to the sample. If they react, they cause the blood to go ahead and clump together. This lets us know that that antibody is found in the blood. So here, we have an example where both anti-A and anti-B reacted, meaning that it has both the A and B antibodies on it. Our second example, only the A type reacted, so this only has the A antibodies on it. The third one, only the B type reacted, only the B antibodies reacted, meaning we have B type. And in type O, the final one, we had no reaction with any antibodies, which makes sense because O type does not have any proteins, A or B, on its surface. So the type that clots with both anti-A and anti-B is AB type. Meanwhile, the type that doesn't clot with anti-A or anti-B is O type. Do you know what your blood type is? If so, that's pretty cool. I'm sorry, that was kind of a bad question to ask. I'm recording. I can't really hear you or what your answer is, but hey, thanks for sharing if you did. The next thing we have to discuss is your blood type can also be positive or negative. And that positive or negative factor is known as the rhesus factor, also known as the rhesus protein. So if you are positive, that means you have the rhesus protein on your blood cells, or if you are negative, that means you are lacking it. Now the downside is if you are Rh negative, you have the antibodies for that in your bloodstream, meaning that you can't accept positive type blood. Now this can be a problem for mothers that are Rh negative or don't have the protein. So they have the antibodies in their bloodstream. And if the baby is Rh positive, that means that the blood cells of the mother could actually try to attack the blood cells of the baby. So what we can do here is the doctor can go ahead and give mom a shot of something called Rogam at about 28 weeks that removes or hides the R positive cells from mom's bloodstream, meaning the baby will be safe. So finally, let's go ahead and do a real problem here. In this case, let's say hypothetically speaking, mom is B type and dad is O type. The baby happens to be AB. And we need to figure out, is that possible? So let's go ahead and figure out mom's genotype. Now we went with BO here because normally the heterozygous condition is more likely, but you could also go ahead and run through this with capital B, if you wanted to. Now considering dad is O type, there's only one combination for that, and that's OO. So let's go ahead and get our Punnett square set up. We'll put mom up top, dad on the side. So in our first box, we would get the combination capital B, O. In our second box, OO. In our third box, BO. And in our fourth, OO. Oh boy. We got a bit of a problem here. So the baby was AB type. This isn't possible with the blood type of the mother and the father. Oh boy. This is awkward. So anyway, that was an example of how to do this. So just remember, you could also run through this one more time using BB as mom's blood type. I mean, unfortunately, you'd still get similar answers, but, you know, you could go ahead and try it. And with that, that's going to go ahead and close up shop. I know we ran a little bit longer in here than the other videos because of the detail about the blood typing. But if you have any questions, please go ahead and contact your instructor. They'll point you in the right direction. As always, thank you for your time. Have a great day, and I'll see you in the next video.